welcome back to the final module in the fungal communities, their dynamics and their interactions with plants, this Jepson herbarium workshop. This last section will be on dispersal of ectomycorrhizal fungi and the limits to forest expansion. So this expands on the last module, which gave you the basics of ectomycorrhizae. Now what I want to talk about is especially how they interact with expanding forest edges and disturbance, and how they manage to get around and colonize root tips. So there's basically two ways to colonize new roots. You can either grow to them by mycelium, or a spore can land near an uncolonized root and colonize it. Between those two, mycelium is much more effective because it can expand further and search for new roots over a broader area. So in this picture, for example, where you see all of this mycelium coming off of this little tiny seedling here, you can imagine that any other seedling that would land in here would immediately run into this mycelium and be colonized. And so that's how mycelium works. And so if the root density is high enough and they're already colonized, a new root that pops up in that area will be colonized by that mycelium. But when no living root or living host tree is present, spores are the only way to establish on a new seedling. And so we'll see this dynamic. And we'll see this dynamic first from one of the very early pieces of work in the field of ectomycorrhizal ecology. This is a review paper that was summarizing some work in the 80s by folks at the Institute for Terrestrial Ecology in Scotland. And what they observed is that in very young birch trees, just a few years old, they would get a set of fungi that they termed the early stage fungi that would fruit near these trees. And as the tree got bigger, those early stage fungi would not disappear, but they would move out to the periphery of the root system to fruit. And that closer to the tree, there'd be a new set of fungi that came in that they termed the late stage fungi. And they would grow closer to the tree. But the most interesting experiment that they did involved putting seedlings under the tree. If you put seedlings under the tree, this larger tree, they would be colonized by the late stage fungi. So this showed that it was not a tree age phenomena that was determining this, right? Because you could colonize these young seedlings with late stage fungi. But if they planted those seedlings under the tree and disturbed the soil first, then they would get early stage fungi. So here's what we think is going on, is that basically, if a seedling establishes in the root zone of an established tree, the mycelium is what's going to colonize it. It's going to grow out. So in this, you could imagine the seedling dropping down here. It's going to be intersected by this mycelium and be colonized by it. And the EM fungi that win this mycelial competition, for reasons that are unclear at the moment, are these late stage fungi. But when a seedling has no access to this mycelium, it's outside this zone, then it can only be colonized by the spore. And the ones that colonize by spores are the early stage fungi. So coming back to this diagram, what that's saying is that spores are not effective inoculum for the late stage fungi. And this has been shown to be true again and again. You can take ungodly numbers of spores of something like Boletus edulis or chanterelles or Amanita or russulas or any number of other ectomycorrhizal fungi, put them on an uncolonized seedling in a pot, and nothing happens. So we cannot observe the conditions under which those spores work, although we know that ultimately they must work occasionally in nature to establish a new mycelium and then start their growth on a tree. In contrast, what we think of, or what they thought of as early stage fungi, and what I'm going to refer to as spore bank species, the spores are effective inoculum, and under these species, you can take the spores, put them in a pot, plant a seedling in, and you get colonization. Although even then, it takes quite a few spores. It's not, it's a numbers game, and it takes many, but they work. So that's basically what's going on here. Is we have these early stage things that respond by spore, late stage things that don't. Resident mycelium on older trees that can then spread out and colonize new roots as they pop up. So a few generalities about spores of ectomycorrhizal fungi. And here I have to say I'm showing you spores of a saprobic or parasitic fungus, mostly because it's a nice picture with white spores everywhere. But it makes the point that spore production is astronomical. You have unbelievable numbers. I'll show you some that are fairly crazy later. But yet dispersal limitation is common in spite of a huge number of spores. And it's because, again, it's a numbers game, that it takes more than one, it takes more than two, because they have to be very, very close to this root tip to work, and probably there at the right time. So establishment by spore is difficult for most species. And as I mentioned, in the ectomycorrhizal world, most species, we can't observe it. A few species establish readily by spore, but these do require host cues. And sometimes we know the chemistry of these cues, sometimes we don't. But they basically sense their host by the chemistry of that root and then germinate. And they have to grow, maybe a 1/10 of a millimeter to get there, and that's a long way for them to grow if it's a tiny little spore, right? So they don't have much chance for it. Okay, so with that background now, I want to walk you through a couple situations here. And the first one I'm going to walk you through is a transect that came from some very early work we did at Point Reyes National Seashore. So what's going on here is each of these circles is a hula hoop sized sample grid where we took 3 soil cores that were about 10 centimeters apiece and went down about 10 centimeters. We'd pull the roots out of that and then identify the fungi that were mycorrhizal on the roots of these mature trees here. And this is bishop pine. And then we'd take the soil from that and plant it into a little pot and put a seedling into it and ask what the bishop pine seedling saw in that soil. And we'd pull out those tips and identify them. And that's this list. And then the transect goes on for a few meters down through this little patch of forest. And so you can see a lot of names that you can maybe barely read or wouldn't know if you did, but what you can see, there's only one that is in red on both. That's the only one that's in common between these two groups. So most of the fungi on this side don't show up on this side. So it's basically like that late stage, early stage paradigm, is that in the soil, we pick up a very small subset of things that are on the mature roots. And these are establishing by spore. The other thing you'll notice here is that there's a sea of blue on the bioassay side, the spore side. And that sea of blue is the genus Rhizopogon that I'm going to come back to multiple times in this talk. Okay, so here is that same data arranged as a ranked abundance curve of species that are found on the mature forest, and then those that were found in the little pine seedling bioassay here from the same soil core. And again, what you can see is that there's not much overlap. There's all these things that are abundant in the bioassay that we can't even find on the mature forest roots. And then there's some that overlap a bit, but are much more abundant in one than the other. So again, what we're looking at here is spores in the bioassay versus mycelium in mature forest. So these are spore bank species. Yeah. Something happened here on my, oh, there we go, oh, back. All right, so with that little background, now let's go and look at the continent and say, okay, what's going on with these spore bankers over the whole area? And so what's going on here is that we sampled across North America, and for the spore bank, we looked in 6 different states, 2 sites at each. And what we would do is walk into pine forests of selected species, sample what was on the roots, and then take that same soil and bioassay it and find out what the seedling saw in the bioassay. So the same kind of experiment I just walked you through, but on a broader scale. And so this is what the bioassays would look like. 12 plots, 13 samples, 2 different host trees that we use, spruce and pine, 1,800 seedlings about altogether. And the results look similar to what I just showed you, in that there's a large number of species that are found on the mature forest roots, and a very small sample that are found in the bioassay, and an even smaller overlap between those two. So again, those that established by spore, less common in the mature forest, but super common when you disturb it. And we look at the geographic pattern of these. The spore banks in similar areas look similar, but across the continent, you get different dominance in there. And what you can see is that this little icon are all the genus Rhizopogon, which again, I'm going to come back to. And we saw in the first one, were dominants in the west and the southeast. So if we go back to this, we could ask the question, what are the spore bank species waiting for? Why aren't they active? And the answer is disturbance. And so we found that answer out because fortuitously, some of the sites burned up in fires after we sampled them. And so here we're looking at one of the samples from that last Continental experiment, and this was in the Sierras in the area of the Rim Fire just outside Yosemite. So this was before it burned, 2 years before it burned, and then here it is after the burn. And when we take soil from the burn and bioassay it and compare it to what it looked like pre-bioassay, it's basically a straight line relationship. That it looks like the spores that were in the soil were not killed by the fire. They were still active in the bioassay, and they responded almost identically to what we saw before fire. If we go and look at seedlings on the ground, as opposed to seedlings in the greenhouse, they too look like the bioassay, with one exception, one that is more common in the forest situation than it was in the greenhouse. And this to work. The 4 most common species here are all Rhizopogons. So here we've seen Rhizopogons point raised setting. We've seen them across the continent now, and they're responding to disturbance in the Sierras. Okay, so one question you might be asking is, how long can these spores wait? And so this is a question that we got interested in a few years ago. And as a result, we set up a 99-year experiment. The intent of this experiment was to get 99 years worth of funding from the National Science Foundation, but of course that didn't work. But it did work for 5 years, and so we've got some interesting data from that. Here's how the experiment worked, is that this is Point Reyes. Along here is the Elk Range, which is devoid of trees by and large. These little flag areas are areas where we sampled the soil and then bioassayed it. And when we bioassayed the soil, we found that there are no rhizopogon spores. In fact, there were no ectomycorrhizal spores that we could detect here. So this area was free of ectomycorrhizal inoculum. That may sound unusual, but it's actually about 11 kilometers away from any forest. So that's, as I'm going to show you, you don't have to get out far from a forest edge before you lose the inoculum for new trees to establish. So this was a free zone of ectomycorrhizal inoculum. So we took that soil and sprayed on 40 billion spores of 3 different species in 3 separate batches, packed them up in flower pots, wired them shut, and buried them back out at Point Reyes in the Elk Range. And then we dig them up once every year for the first 5 years, and then once every 5 years after that, and now we switch to once every 10 years. And we've managed to get a few papers out of this now. The last one just came out from the 15-year burial experiment. And we picked up year 20 a month ago. And we haven't set up the bioassays for that yet. This is the way that we then analyze that data, is we take the soil and dilute it in a series of twofold dilutions, and then plant seedlings into it and look for colonization. So what's happening here is each of these is a twofold dilution. And up here, there's so many spores that 100% of the seedlings are colonized. And then you hit a spot where, okay, these fewer than 100% start to get colonized, and then more dilution doesn't matter, nothing's colonized. So it's a sigmoid curve is what we see from this. And we saw that for all 3 species that we did. These are all rhizopogons. And the way we're going to compare these is the interpolated concentration at which 50% of the seedlings get colonized. And that's important because otherwise you have to compare graphs that look like this. So this is all 7 time points for those 3 species. And you can see you get sigmoid curves over the whole realm. And you've got an eye for color. You can see there's some pattern in the coloration which corresponds with the years. But the easiest way is to look at those C50s. And when you do that, it looks like a loop. So what's going on is that for the first 4 years, it took fewer spores to colonize 50% of the seedlings than it did at year 0. So I'll say that again, because it's counterintuitive. It took fewer spores to colonize as the years went on. But then at some point, at an unknown inflection point, it starts to curve back up and starts to again take more spores to colonize 50% of the seedlings. So what we think is going on here is that it's basically a changing balance between dormancy and mortality. That what's happening here is we're breaking dormancy and recruiting some of these spores into a population that can then respond to these roots. And at the same time, there's mortality going on, so we're losing some spores, but the rate of dormancy break is greater than the rate of mortality early on. And then at some point between year 5 and 10, it shifts, and we start to see greater mortality than dormancy break. So the quick answer is that they last at least 15 years. And from these trajectory, we could predict that it'll be a few decades before we lose colonization potential of these spores. So what we know, what I just showed you was that spore banks are widespread and dense in these pine forests. They're relatively simple and dominated by a few genera. The dominants vary geographically. They're very important post-disturbance in fire. I could have shown you a couple other fire examples, and also logging. Some species have been shown to last at least 15 years in the soil, and multi-decadal longevities are likely. Okay, so let's go back out to Point Reyes, because it's a great place to look at the patterning of these colonizations. And the reason it's a great place is that when we go to a pine patch like this, it's all mycorrhizal. But when we go out to this coyote bush, poison oak community out here, that's all non-ectomycorrhizal. So this is actually an edge. And we can ask, what does the inoculum for Ectomycorrhizae look like out here? So for this, we'll turn to some recent work from Kabir Pei's lab. And what he did was collect soils across distances from established trees, going from right next to them out to about 1 kilometer, and then take that soil and bioassay it and say, okay, who's there and how much do we see? And when he does that, we get some different curves with different species. So you can see this is a log scale down here. So you'll see this is only 10 meters from the forest. And some of these plateau within 10 meters and then are just not seen again. Here are the rhizopogons. We'll come back to each of these, but let's look at this one first. It really didn't get anywhere. This is Tomentella, one of these little crust fungi. Here's the little piece of Tomentella that you're looking at here. So it's not much. So you can see why it's not going far, in part because there aren't that many spores, right? And they're not getting released to the air very effectively. But the other reason is that it's probably moves more by insects than it does by air. And Elsa covered this a little bit in the introduction, but here are mites, and we see these dark blobs in them. If you blow those up, those are blobs of these knobby spores of Tomentella. This is what the frass from those mites looks like. It's just loaded with those spores. If we stain them with FDA, which tells us whether they're living or dead, a bunch of them still look like they're living. We can find them in Colembula too, which is another little microinvertebrate. We can find them in millipedes. We can find them in beetle larvae. We can actually find them in salamanders that have eaten these. And when we take those spores and put them onto seedlings, it will cause colonization. So these look like they kind of go through the microinvertebrate food web. And so it's not surprising that they're not getting far from the forest edge. How about the rhizopogons? If we look at the rhizopogons, they too plateau reasonably early. They make it about 100 meters from a forest edge, and then their frequency goes way down. How do they move? Well, there's some hint of it here. You can see some little gnaw marks on here, and that's because mostly they move by small rodents. That these are little packages of spores, truffle-like packages that are eaten by small rodents and then defecated out in their scat. So to look at colonization of these, what we did was put out little rat feeding boxes, but instead of poison, we put in oatmeal and just attracted in little rodents and then collected their dung and put them around at various points in Point Reyes, either in forest or outside of forest, either in burned or unburned areas. And so now we get to the quiz part of this, right? This is when we find out whether you know your shit. This is, of course, mouse or vole. This is rat or squirrel. And the other one that was in the box, we were scratching our heads over for a long time and then realized we're looking at banana slug. So the banana slug here has reprocessed the rodent pellets. But in any of these, when we looked at them, if they were within a few tens of meters of forest, they were just loaded with spores of rhizopogon. So that's what you're looking at here, is just a sea of rhizopogon spores. So very effective dispersal, but for a short distance. So you can see here it is over just 30 meters from the forest edge. It's already starting to drop. And after about 100 meters, it's gone. You get it sporadically out further by deer, which also eat these, but they're not as common of a vector. And you'll notice there's a difference between burned and unburned forest edges. And that's because when you look at burned forest edges, there's a much greater density of rhizopogons being produced under these young trees. And so there's more dispersal going on when the forest is young and it's establishing the spore bank. And then as the forest matures, there's fewer rhizopogons being produced and less dispersal. And Point Reyes, it kind of looks like this, where we've got a couple of these species that are found only in the young forest, and then we've got a couple that either start there and peter out or show up later. So there are rhizopogons at all levels, but some different species involved. Okay, let's go back and talk about Suillus. So Suillus, if you look at this, is the one that really gets out there. Here we are a kilometer away, and it's getting about 25% of the seedlings out there. So it's not 100%, but it's better than anything else. And the reason it can do that is it's a big mushroom. It fruits in huge quantity, and it produces a lot of spores. So to measure how many spores and how effective that was, we put out uncolonized seedlings out into the coyote bush here at various distances from trees, and also collected rainwater here. And in the rainwater, we could spin it down and quantify the Suillus spores that were in it. And what that looks like is this, is that it drops rapidly. Again, this is a log scale. So by the time you're out a kilometer, there's about 100 spores per square meter that get out there. But when you're back near the forest edge, it's more like 1 million. So there's a lot of spores. And if you model this, which is what Kabir Pei did, you see that around the forest, the amounts are again around 1 million spores per square meter per day. So that's a lot of spores. And by the time you're out a kilometer, it's dropped precipitously, but there's still a lot of spores, and yet only about a quarter of the seedlings will get colonized out there. So we're hitting this level where, even though we have this astronomical level of spores, the forest really can't expand that fast because it's limited by them. And we could see this again in other settings. This is England, some work by Collier and Bidartando, where they're looking at invasion of heath by ectomycorrhizal trees. And so back in the forest, you can see there's a huge list of ectomycorrhizal fungi associated with these trees. When they look at the heath that's being invaded and look at the species on that, that's a much smaller list. And then if they go off in the uninvaded and put seedlings out there and ask what can colonize them, it's a very small list. And that list for pine is basically Suillus and rhizopogon, especially Suillus. And in their conclusion, they were saying that a small set of ectomycorrhizal fungi mediate this tree invasion as a result. And I should emphasize that small set is Suillus. And tree seedlings could remain non-mycorrhizal for at least a year when no inoculum was present, but they didn't grow. And when they become mycorrhizal, they start gaining biomass. Now, the last setting I want to move to is the southern hemisphere. And in the southern hemisphere, there's a big problem actually with pine invasion. Pine is not native in the southern hemisphere, but it's really taking over in some spots now. And this is one of such spots. This is Cayoque, Chile. And you can see this just 8 years apart. Pine plantation here, now it's spreading out this way. And now this is all self-planted pine. This is a color picture of that same general area with the pines establishing in what was a grassland at high altitude in the Andes. This is, and I should mention, this is lodgepole pine, our native pine. Now, what drives this is the mycorrhizal fungi. And what Hayward et al. Did was to sample this community, and they found that as they sampled more and more and more trees, they did not find more and more and more mycorrhizal fungi. They found 4 in total. And really, there was just one that was key. That one was Pseuilus. And this Pseuilus was a European Pseuilus that was moved accidentally into Chile and is now associated with our lodgepole pine. And in those that were any distance from the forest edge, it was always Pseuilus that was associated with it. And in fact, Pseuilus was detected associated with every tree that they sampled except 5. And the 5 trees which did not detect Pseuilus were the ones closest to the plantation edge. So close to the plantation edge, other things, some of these other 3 that were there started to associate with it and outcompete the Pseuilus. But as you got away, it was all Pseuilus, and you can see Pseuilus fruiting under it here. So the take-home message on dispersal of ectomycorrhizal fungi and the limits to forest expansion are that the most effective way for EM fungi to find a new host root is by mycelial growth. This is what's going on in a closed forest system where there's no disturbance, right? This is the early stage, late stage paradigm. It's going to be those late stage fungi that grow out and find a new uncolonized root. Spores of most ectomycorrhizal fungi do not germinate under observable conditions. A small subset of these have reactive spores, and these are the same ones that colonize seedlings after disturbance. These are the ones that are calling the spore bank fungi. Ectomycorrhizal forests cannot expand into areas lacking ectomycorrhizal propagules, and it's the pseuiloid fungi that really drive the forest expansion, both here and in Europe and in exotic settings like Chile. So with that, we will end this session on ectomycorrhizal fungi and limits to forest expansion, and we'll end the Jepson it will end the Jepson session as well, and I will take any questions.